In the previous video, we learned about what is post-transcriptional process, the types of modifications involved in the post-transcriptional processes, which includes 5' RNA capping, 3' polyadenylation, and splicing. We then also looked to the mechanism of capping and polyadenylation along with their specific importance, and then we finally learned about the third post-transcriptional modification, that was splicing. Now, in this video, we will continue with the splicing process. So let me again define what is splicing. So splicing is defined as a highly precise removal of intron sequences. So in this figure, you can see your intron is being removed in a form of a lariat structure. And how exactly this lariat structure of intron is being removed? It occurs by the two trans-esterification reactions in which the phosphodiester linkage within the pre-mRNA are broken and the new ones are formed. So the first reaction is triggered by the 2' hydroxyl group present at your A branch site. This group acts as a nucleophile and attacks the phosphoryl group of the conserved G in the 5' splice site. As a consequence, the phosphodiester bond between the sugar and the phosphate at the junction between the exon and intron is cleaved, and the freed 5' end of the intron is joined to the A within the branch site. Thus, in addition to the 5' and the 3' backbone linkages, a third phosphodiester bond extends from the 2' hydroxyl group of that A to create a three-way junction. Now for the second reaction, the 5' exon, or more precisely the newly liberated 3' hydroxyl group of the 5' exon, reverses its role and becomes a nucleophile that attacks the phosphoryl group at the 3' splice site. This second reaction has two consequences. First, and the most importantly, it joins the 5' and the 3' exons together, and secondly, it causes the removal of intron in a form of a lariat structure. So this figure again shows the molecular mechanism of splicing, which involves the two trans-esterification reactions for the removal of intron in a form of a lariat structure. In the two reaction steps, there is no net gain in the number of chemical bonds, that is, two phosphodiester bonds are broken and the two new ones are made. It's just a question of shuffling on bonds, and there is no energy input which is demanded by the chemistry of this whole process. So now let's study about the spliceosome machinery. The trans-esterification reactions that I told is basically mediated by a huge molecular machine which is known as a spliceosome. So this spliceosome comprises about 150 proteins and 5 RNAs, which are similar in the size of that of a ribosome. In carrying out even a single splicing reaction, the spliceosome hydrolyzes several molecules of ATP. The 5 RNAs which includes in this are U1, U2, U4, U5 and U6, which are collectively known as snRNAs or small nuclear RNAs. Each of these RNAs is between 100 to 300 nucleotide long and is complex with several proteins. These RNA protein complexes are known as snRNPs or small nuclear ribonucleoproteins. These small nuclear ribonucleoprotein or snRNPs have three different roles in splicing. First, they recognize the 5' splice site and the branch site. Second, they bring those sites together as required. And third, they finally catalyze the RNA cleavage and the joining reactions. So let's study about the splicing pathway. Initially, what happens, the U1 snRNP recognizes the 5' splice site because of its base pairing between its snRNA and the pre-mRNA. One subunit of the U2AF binds with the pyrimidine trap and the other binds at the 3' splice site. The former subunit interacts with the BBP protein, ensuring its binding to the A branch site, resulting to produce a complex known as E complex or the early complex. This early complex gets converted into the A complex by the action of the U2 snRNP, which displaces the BBP protein and binds itself at the A branch site. Now, this A complex undergoes a conversion to B complex, which occurs by the action of the tri-snRNPs. The tri-snRNPs includes U4, U5 and U6. So as you can see in this figure, as these tri-snRNPs comes and bind to your pre-mRNA, it tends to fold your pre-mRNA strand, and they also displaces the U2AF65 or 35 subunit from the 3' splice site. Now, this A complex undergoes certain rearrangements to produce a C complex, which is known as an active complex. Therefore, you can see during the rearrangement, it replaces or displaces your U1 snRNP first, whose position is occupied by the U6, and then the U4 snRNP is removed, whose position is occupied by your U5. Now, this complex is very much active for undergoing the process of trans-esterification reactions that I told, and it results to the removal of your intron in a form of a lariat structure, and all the exons are spliced together. So let's study again in a simpler manner the splicing process. So this is your 5' exon and this is your 3' exon. Now firstly, what will happen? The U1 will come and bind to the 5' splice site, that is the intron exon boundary, and then the U2 will bind at your A branch site. Then another complex, that is the tri-snRNP complex, will come and bind to this pre-mRNA strand, resulting to the folding of your mRNA as shown here in this diagram. Now this spliceosome undergoes certain rearrangements due to which your U1 and U4 subunits are removed. Overall, your pre-mRNA is occupied by U6, U5 and U2 snRNP. Now, there occurs a close proximity for your 2' hydroxyl group present at your A branch site to act as a nucleophile to attack the phosphoryl group present at your 5' splice site. Therefore, it results to the first trans-esterification reaction, and the second trans-esterification reaction occurs by is mediated by your U5 snRNP, which causes the cleavage of this phosphoryl group present at the 3' splice site. Thus, you get a 5' and a 3' spliced exon with a lariat structure of intron and all your snRNPs out. Now this lariat structure can be converted into a linear form of an intron RNA by the action of a debranching enzyme. Always remember that not all the introns are completely degraded. Some end up as a functional RNA, which is different from mRNA. Only after all these modifications, the mature mRNA leaves the nucleus and enters the cytosol for the process of translation. All 5' capping and 3' polyadenylation addition gives your mRNA molecule the ability to resist the degradation by different types of enzymes or proteins like DNAs and RNAs, and it also gives the mRNA the ability to travel from the nucleus to the cytoplasm of our cell. These were my references. Thank you.